Oh, that's a good question. It's always questionable. All right, so first big thing is hopefully you saw the letter from the chancellor that came through last night, that we are going back to face-to-face next week. So our very next meeting is going to be at Cypress in an actual room. And so we're going to have face-to-face classes from here on out, and we will have lab in the actual lab room. So that's pretty exciting. So I'm still kind of figuring out what I want to do with that. I haven't had a chance to really think about it, to be honest, but we're going to continue with our lessons. We're going to finish up chapter 1 and hopefully start chapter 4 next week. And then, you know, our tests won't start until the following week. So we'll have a little bit of time to kind of get our feet together, which is good. But as far as things that make it different from what we're doing, obviously, we're not going to be meeting on Zoom, I assume. We'll be meeting in an actual room, and I don't remember the room number. I'll have to look. It's in the syllabus, right? It's in the syllabus, and we'll all be there. Do need to be on time. And a couple of things to be aware of. One, masking is required, okay? If you're anti-mask, you don't have a mask, get a mask, okay? To me, it doesn't seem that weird because, you know, when we do lab safety stuff, we require lots of clothing things. You know, you have to have closed-toed shoes, you need to have long pants, you have to have safety glasses depending on the activity that we're doing. And so wearing a mask in addition, even though it's kind of a pain, right? I don't like wearing them. It's not that big a deal to me to require another clothing thing, right? So just think of it that way. We're using lab safety rules for all our classes on campus. So get a mask, plan to bring it, plan to have it on, not like under your nose or like hanging on your ear, you know, like, but on your actual face. And we'll just wear those. I'm not looking forward to teaching in a mask, but I'll deal, you know, if it helps make us safer, which based on the evidence I've seen, it really does help. I'm gonna do that, okay? We will respect social distancing as much as we can, okay? We'll see what we can do. The room we're gonna meet in is actually a computer lab, so we'll have computers in there that we can use for various things. But we'll be doing in-person interaction as well, okay? I'm gonna work on exactly what I want to do and what you should expect. And I'm going to type that up in a big email and send it out over the weekend so that we're all nice and organized. You guys know what to expect. So look for that. Okay. All right. Questions. Let's see. I see names. Any questions about that? Oh, other things. It is likely that they will ask you for an ACC ID when you come onto campus. There will be only certain doors that you can come in to, and there will be like a check-in station that you have to go through to get on campus. At the check-in station, at least the way it was, excuse me, during the height of the pandemic, they would check your temperature. You have to use a little app. You have to fill out a little form, and I can send you information about that. That says, you know, what symptoms do you have, etc. And once you filled that out, that just confirms, you know, that you're saying, and I don't have any of these symptoms. I believe they're going to check IDs as well. And then once you get all that done, then you can come onto campus. So please allow yourself a little bit extra time to do that, because it's probably going to be kind of a mess the first couple of days with everybody figuring it out. And I don't want you to be late for class. Okay. I can stall for a couple minutes at the beginning of class, but not a whole lot. So plan to get there a little bit early to deal with that stuff. Okay, and I have to do it too. Okay, questions.
So next Tuesday will be for sure in person?
Yep, next Tuesday in person. If you don't have an ACC ID, I did include information on the syllabus. If you go to our course syllabus and type in ACC ID, it'll take you to a section and it'll tell you where to go online to apply for one and to get one.
I have an ACC ID from like 2 years ago. Is that one still okay, or do you think?
Yeah, that should be fine. Yeah, that should be fine. Okay. Seeing what else happening with our participants here. So if you don't have an ACC ID, I'd say just get one anyway. If you're going to take ACC classes at all, it's a handy thing to have, right? Well, you have one of those mics. I do. I did. I planned a few weeks ago and I got one of those microphones. I don't know how well it's going to work. We'll try it. In some rooms, it seems to give me feedback, which is a no-go if there's a bunch of feedback, but we'll try it out. We'll see how loud I need to be. I can be pretty loud. Yes.
Wait, so we're going to be doing 2 different rooms, right? Like it's going to be in one room, the computer lab is where we do the lecture. And then in the other room is where we do the actual labs.
Correct. Correct. And they're right across the hall from each other. So as long as you find the first room, we can find the second room.
Are we still going to need to do the watch the lectures beforehand, or is that just something we do because of online? I was kind of confused about that.
Nope, we're gonna conduct class the same way. The only difference is that when we meet and do activities and lecture, we'll be together in a room instead of on Zoom. So all the same expectations. There are videos that I want you to watch beforehand. And some of that is just a time thing. That gives us the time to do activities and things. The nature of our activities will change a little bit because we'll actually be in person. Okay. And I'm going to have to think about what kinds of activities we can do that will still kind of respect social distancing and things like that. So we'll do the best we can. Okay. Yes, you do need, you're coming to class on Tuesday, face-to-face on Tuesday, correct. Another thing to think about with this, and this is going to be kind of weird and a little bit different. If at any point during the semester you wake up and you feel ill, especially if you have any of the COVID symptoms, and COVID symptoms are pretty common for a lot of things, right? But anything like fever, a cough, tightness in the chest, I assume lots of sinus, that kind of a thing.
Also,
Then,
sorry, just want to throw this out there. If you have kids or if you're around kids, I heard that when I went to the doctor, they said that most of the children's are showing as gastrointestinal problems. So watch out if you have gastrointestinal issues too.
Oh, weird. it's just a mess. I'll see if I can compile a list of symptoms. But if you're waking up and you feel ill, right, don't just try to power through it and come on to class. And that's going to apply to me too. I'm just as bad about that as y'all are. Like, I'm just going to, go to work. And I have definitely done that in the past, but I will work with you guys if you need to miss class because you're ill, but don't take a risk of coming and spreading something potentially bad to everybody else. Okay, we need to be extra careful about that. We're going to see how that plays out. Someone have a comment?
Ms. Farrell, yeah, I had a question. If we've been vaccinated, should we bring our card?
They're not going to check whether you're vaccinated or not. We're not allowed to ask. Y'all are welcome to share that info with each other if you want, but they're not going to check for that.
So if we do wake up feeling sick, do we just like shoot you an email?
Yes. Yeah, that's the best thing to do. And then, I actually have all of my lectures recorded. So almost all of them. So if you need to, I could send you links for those. And that's one way to kind of make it up. Now, if we're meeting in person, I'm not going to have a camera at the back of the room recording me doing that. So that's one feature that we lose going face to face. But I do have lectures that you can watch. Okay, and I'll be flexible with that sort of thing. So just be honest about it. Don't lie and try to come to class anyway. If you're ill at all, stay at home. Okay. So we all get to play the fun. Do I have allergies or do I have COVID-19? Right, this semester, which is very unfortunate. It'll be okay. We're gonna work around it.
And do we need to bring like a doctor's note if we were sick and we like went and got tested and everything, or is it just like on us?
That's just on you. I don't, as far as disclosure, if you were, I know that the school is going to offer COVID-19 testing. So if you are suspicious, you think maybe you have it, you can go and get tested. I think at the campuses, I think Highland, I don't know about Cypress anyway. But you don't have to disclose that if that's the case. Yeah, if you did have COVID and you recovered, you don't have to disclose that. Okay, I know it's complicated, right? My big fear is that someone finds out they're COVID positive, comes to class, exposes everyone, or God forbid if I do it, because I'm going to be around even more people than y'all are, then we all have to quarantine kind of a thing and go back to virtual. And I'm hoping it'll be fine. Hoping it'll be fine. Okay. We're going to play it by ear, y'all.
Some people don't show symptoms though.
Right.
So
That's the other part of the problem, yeah, is
Yeah.
that if you're asymptomatic, you can still spread it. So hence the masks, and we're going to do a lot of cleanliness stuff.
Ms. Farrell, I had a question about the lab attire. So instead of wearing goggles, could I wear like a face shield?
You could wear a face shield.
Cool, awesome. Thank you.
Face shields make it hard to hear people, but you could wear a face shield. That's acceptable.
I have one because I'm a dental assistant, so I have those. So I like those more than like goggles, but I guess it depends on the lab too.
Yeah, yeah. We won't have to wear goggles that often. You'll need them for a couple of days of dissection. And that's about it, because we already kind of went through the wet labs that have chemicals and stuff. But I will let you know beforehand if you need to, what PPE you need to wear for that day. Okay.
Yes.
And we're gonna need to
Thank
buy
you.
the PPE.
I'm sorry?
And so are we gonna need to buy the PPE ourselves?
You'll need to buy the lab goggles, yes, or glasses. And that's in the syllabus. Under things to buy. But I'll remind you, of course. Okay. Other questions about that? It's kind of exciting. I'm looking forward to seeing you guys. Right now, you're just like a field of names that I kind of vaguely know. It'll be really nice to put at least part of a face with a name and put voices with names. That's going to be good.
Is the testing center still going to be closed? We'll be doing exams in class, right?
You'll be doing exams in class.
Okay.
Yeah.
Thank you.
Yep. Yeah, testing center, it's not closed, but it's very limited use. We have to get special permission to use it now, so. Okay, any other questions? And again, I'll, my emails, I'll type it up and put it in an email.
So if we're sick on a test day, how would that work? Is it gonna be like in the testing center?
We'll figure it out. We'll figure it out. You'll get to take your test. I would probably have you do it online. I don't know. I have to think about that. It's a good question. All right, anything else? All right, y'all.
Question, sorry.
One more, okay.
Yeah, are we doing our tests on computers, or are we gonna have like paper tests?
The tests are gonna be computerized.
Yeah.
But we have computers in the classrooms. Okay, okay. All right. So without further ado, let's get into, we need to finish chapter 3 today. And I want to dig into chapter 1 and do as much of that stuff as we can. So with chapter 3, we're going to be talking about membrane potential today, and we're going to be talking about cell communication. And then we're going to jump over in chapter 1 is going to have some totally different topics, some things like homeostasis, and hopefully we'll have time to start talking about body regions and stuff like that. Okay, so we've got a variety of topics to tackle today. Here's where I want you all to start. I'm actually going to start you with a group activity to review what we did last time. I added some things to the worksheets that we used last time. So if you remember, you were using these group worksheets to answer questions about the lab. And the lab quiz is now available. I finally had a chance to sit down and get it. And sorry it was late. But the passive transport lab quiz is now available in the chapter 3 folder. It's not due until Sunday night by midnight, but if you want to go ahead and knock it out, you can. Okay, and that'll be based on this info that we went over last time. Okay, at the bottom of that sheet, below that section about passive transport lab, there is a nice big table here that allows you to compare the different kinds of transport that we talked about. Okay, we're comparing simple diffusion, facilitated diffusion, both of which are passive, and active transport. And so what I want you guys to do is work together in your groups to try and fill out this table. Now, if you want to try and push the table down onto one page, you can. You guys can adjust this however you feel like. There we go. That's kind of better. Okay, but do whatever you need to do. Okay, these are your working documents. Again, you're not going to have to turn these in, but I want you to see if you can fill them out, think about it, and we'll go over them together. Okay, so I just want you to do that section first. And then where it says pause here, pause there, because we need to go over some more material. All right, so let's start with that. I'm going to just randomly put you in groups. You may be in a different group than you were in last time, and that's fine. These are just things, documents you can use to collaborate with one another, right? So what I'd like to do is go over this chart with y'all. And what I'd like to do is, instead of calling on people, is have people from different groups volunteer to talk about this a little bit. And so let's go over it together. All right. So first, we're making a comparison of simple diffusion, facilitated diffusion, and active transport. And this table's a nice way to just kind of summarize what you know. All right, and I'm in group ones right now. And so the first thing it wants you to answer is, do these methods move molecules with or against the gradient? In group one, you guys put no and yes, which doesn't really answer the question. So I'd like to ask someone from group one. So does simple diffusion, does it move things with or against the gradient?
With.
With, you got it. I assume you meant no, not against the gradient. Oh, caps lock. There we go. All right. Thank you. How about facilitated diffusion?
With.
With as well. And active transport?
Against.
Good. And when you say with the gradient, what do you mean? From what to what concentration?
High to low.
High to low. Got it. So high to low is with or along the gradient is going to be high to low, if you hear those terms. And the opposite can also be true. If you have low to high, low concentration to high concentration, that's against or up the gradient, sometimes it's called up the gradient or against the gradient. Okay, good. So you can tell these two passive methods move things high to low. That's what makes them passive, whereas active moves the opposite way. Very nice, thank you. All right, let's see. And the second one, move molecules into the cell, out of the cell, or either. And you guys put yes or no here as well for a couple.
Oops.
Uh-oh. If you hold down the control button and hit Z, it'll back you up a step. There we go. Awesome. Okay, so Jason's fixing it there. So move molecules into the cell, out of the cell, or either. So with simple diffusion, correct? They can move either direction. It just depends on where the gradient is. What about facilitated diffusion? Also both, yeah, and active transport, also both. So there's no particular direction into or out of the cell. It depends on the specific situation for all three of these kinds of transport. All right, very good, thank you. All right, uses a protein. Let's see what group two's got going on here as well. Okay, so uses a protein. Correct. No, yes, yes. Now, when we're talking about proteins, someone from group two tell me, when you're talking about using a protein, what kind of proteins are we talking about?
A channel protein and a carrier.
Channels or carriers, you got it. Also known as transport proteins. All right, channels or carriers, also known as transport proteins. There is a specific name for the type of protein that we find in active transport. Someone from group 2, do you remember what that is?
Potassium pump.
Say again?
Potassium pump.
Pump is the word I was looking for there. Right, if you see the word pump, sometimes they're also called carriers. Channels, it depends on the channel. Carriers definitely, but if you hear the word pump, pump is going to refer to active transport. All right, very good, thank you. Let's see. Now, these kind of confused y'all a little bit. Requires energy, requires ATP. When I say requires energy, I mean any form of energy. Is any form of energy required to make this transport happen? And so group 2, you said yes, yes, and yes, yes, energy is always required to some degree to make molecules move, right? Now group 2, could you maybe tell me what kind of energy we would be talking about here? What kind of energy is always required to make things move?
Kinetic energy.
Kinetic. I'm going to add that note. Okay, kinetic, kinetic, kinetic. Molecules are always moving, they're never sitting still. The only time they would ever completely be sitting still would be if we reached a temperature called absolute zero, and that's like a theoretical, yeah, we won't get into that debate, but a theoretical situation that we don't see happening in nature. So molecules are always moving, got to have some energy. All right, let's take a look at group 3 over here a little bit, and you guys can be adjusting these as you go if you want to have this as a study sheet for later. Does it require ATP? Dun, dun, dun. Group 3, why? You're correct, but why doesn't simple diffusion and facilitated diffusion require ATP? Why do we not need it there?
It's just going with the gradient. The kinetic energy is enough energy to get it past the membrane. There's no real need to have ATP involved.
You got it. You got it. Yep, because it's moving with the gradient, there's no extra energy involved. So active transport's the only place you need that. Very nice. Thank you. Let's see. Is it specific for certain kinds of molecules? What do y'all think about this one? What did I mean by that? I would say, I'll just kind of give you the answer I was thinking of for this one. I would say that facilitated diffusion and active transport are definitely specific for certain kinds of molecules. And the reason is that they are going to be using because they use transport proteins. And remember that those transport proteins, they're gateways, but they are specific gateways. They don't just let any old body through. Only certain molecules can go through those gateways. So these two are very specific for what they can let through. I would say that simple diffusion, I would say no. Because all molecules really like to move from high to low concentration. Now, whether they can go across the membrane or not, that's another story, right? But I would say no for that one, for simple diffusion. That's not particularly specific, as long as things are moving according to a gradient. Okay, let's see. Rate limited. That means, is there anything that can slow it down, or can it go just as fast as it feels like? And I agree with group 3 here. No, it's not rate limited. And the other two are. Now I'm going to pick on group 3 one more time. Oh, I haven't picked on group 4. Let me look. I knew I was forgetting somebody. Correct. Okay, I'm going to pick on group 4 then. Can you tell me why? Why is simple diffusion not rate limited but the other two are. Who's feeling brave?
I put no for the simple diffusion because I was thinking it was just it was from high to low with the gradient, so it's just, you know, kind of making its way in and out.
Mm-hmm. And where is it making its way in and out?
The membrane, like across.
Right. Because molecules can cross any portion pretty much of the membrane.
So I originally was maybe thinking if that had to do if the molecules were like hydrophobic or hydrophilic,
Mm-hmm.
but this would be something a little different too, because these are all the different proteins, correct? Well, not all of them, but.
Right, right. So, I mean, there are some things that can affect how quickly they can go across. Well, let me think about that. They can go across pretty much any section of the membrane if they are diffusing. Some are going to be faster than others. That's a good point. Let's note that. Polarity and size.
Yeah, we also talked about the surface area, how the surface area in the membranes in the lungs, for example, will be faster than others.
You know, that's a really good point. That's a really good point.
It is.
Yeah, that's what kind of we went over in our group too for the first one. That's like we didn't know what kind of rate, because there could be that rate and then there's the speed depending like what kind or size.
We were like really thinking outside the box, like
That's great. I love it. Surface area available and limit. Okay. So let's change it. I agree with you guys more. Oftentimes when you look at sources, they will say that diffusion is not rate limited compared to the other two kinds, but you're right, it is, but it's less limited, let's say. Less limited than the other two types. Okay, because it doesn't have to go through a protein. Right, so with facilitated diffusion, we're talking about channels and carriers, and with active transport, we're talking about pumps, and those can only let things through so quickly. Only so many molecules will fit through them at a time. So you can think of it as being like, you know, are you going into a facility and you can enter through just any door? If it does have 1,000 doors and you can go through any of them, or is there just one door and you can only go through that one? If there's just one door, it's going to slow it down. So let's say through transport proteins. Good. Excellent thinking, y'all. I love it. All right.
What does that one say? The one that you just copied?
No, I said because molecules must travel through transport proteins. Because molecules must travel through transport proteins. That slows them down. All right, let's see what examples y'all came up with. Okay, excellent. So simple diffusion would be things like water, oxygen, CO2. See, facilitated glucose is a good example. Amino acids. And sodium and potassium pump, good example of active transport. Excellent, y'all. Let's see if there are any other interesting examples here.
Would that also be considered with the synthase pump? Is that correct?
Synthase. Oh, you mean the ATP synthase?
Yes. That's a
Let's
pump,
think about
right?
that. That's not a pump. It has a channel built into it.
Okay.
ATP synthase might be an example of facilitated diffusion.
Okay. Oh, yes.
Yes, if I was going to classify that one, I would say that. Yeah. Glucose. Yep. Y'all have those similar ones. Let's see, just here, just to see. Group 2 molecules across any portion. Oh, wait, I wrote that. There we go. Osmosis, water, oxygen, good. Glucose. Oh, I see some typing. Potassium, oh, good. And there's going to be lots of others that we'll encounter. I just wanted you guys to think of what you've seen already.
Is this gonna stay on Blackboard?
Yes.
Cool.
Yeah, it's already posted under the chapter 3 folder.
But with like everything because I was like writing some stuff, but I just like, I couldn't keep up, but I liked how we're adding things.
Yeah, I like that too. Yeah. Yeah, anytime we do a shared Google Doc or any kind of thing like that, and we may still do some, even if we're meeting face to face, but anytime we do that, I will post the links on Blackboard under the chapter to which it
Awesome.
applies.
Thank
Mhm.
you.
Yep, absolutely. Great. Okay. Anybody have any other questions about passive and active transport?
I'm a little confused on the middle one. Let me see. What's it called? The facilitated transport. Could you just give like a brief explanation of that?
Of what it is?
Like,
So
like an overview.
yeah, so let's think about the word, and y'all should know this. What does facilitated diffusion mean? What is diffusion?
Is going through like a membrane.
Okay, so it's molecules moving from what to what?
Working for each other.
Concentration from high to low.
So it's diffusion is molecules moving high to low. Sometimes with a membrane, sometimes not. That's just diffusion in general. And then what does it mean to facilitate something?
Will allow it to happen.
To allow it or to help it. Yeah. So it's diffusion with a helper, and the helper is going to be a protein. And that's something that we can add kind of there is simple diffusion is usually for, well, it can be for anything. I would say any molecule. Facilitated diffusion. Well, no, not exactly. No, I'm not going to complicate it. I'm not going to complicate it. Never mind. going too far with it. I was thinking that facilitated diffusion is really necessary for molecules that have charges or are really large. Those are the kind of things that you need a helper for. Okay, good. Any other questions about that?
So would that also consider like a leaky channel?
Yes,
Okay, cool.
as long as it's moving something from high to low.
Okay.
A leak channel would definitely be facilitated diffusion. A leak channel is just an open door, right? So the only way molecules would move through an open door would be from high to low concentration.
Mm-hmm.
Okay. All right. Well, without further ado then, let's move over to chapter 3, part 3, and finish out chapter 3. It's a dizzy. Chapter 3's got a lot of stuff in it. And keep this in mind when you guys eventually, get into ANP2, you're going to talk about these transport mechanisms a lot. Especially, starting out right with the endocrine system, talking about hormones and stuff. They all use these kinds of mechanisms to travel and get into cells and all of that stuff. So when you get into ANP2, keep these notes so that you can refer back to them. Remember to come back to chapter 3. All right. So there are 2 portions to chapter 3, part 3, talking about other membrane functions. And so these are some other things that membranes can be used for. And there's 2 kinds of parts to it. We're going to talk a little bit about membrane potential, which is something I asked you to review before class. And then we're going to talk about how cells communicate using their membranes. And then there's a third thing I forgot about. We'll talk about some connections, some things that allow cells to connect together, some terms that you'll hear as we go through different organ systems. All right, so I'm not going to lecture all these slides because, again, I asked you to watch the video before class. But we're talking about membrane potential here, which is a separation of charge across the membrane. In this lecture, I talked a little bit about how it can be measured, what the typical membrane potential is for an average cell, and kind of how it gets established. And so since I asked you guys to watch this before class, I have a few questions for you. All right, let's do one of these. I'm going to be quiet for about a minute, and then I'm going to call on some folks, and hopefully you did the homework and took some notes on this. And let's see what you guys can tell me about it. And again, if you make mistakes, this is okay. This is the place to do that. All right, so I'm going to be quiet for about a minute. All right, y'all, that was about a minute. I'm just gonna go to my list. I'm gonna pick on AJ first. So the first question up here is, what is membrane potential? AJ, how would you define it?
It's the difference in electrical potential energy between the inside and the outside of the cell, as divided by the membrane.
You got it. You got it. So the membrane's a barrier, and we can have a difference in the charge outside the cell versus inside the cell. Now, what would provide that charge, y'all? I'll open it up to the class. What kind of molecules or what kind of particles would provide those charges?
Electrons.
Ions, right. Electrons could as well, but we don't usually have electrons just floating around because they can be kind of dangerous. Yeah, ions. So when we have different concentrations of ions in different places, it can create this electrical difference too. So an electrical gradient, okay? Has the potential to do work, so we call it the membrane potential. Very nice, thank you. All right, I'm just gonna go down my list. I'm gonna pick on Ariel next. Ariel, can you tell me, how do the charges inside and outside a typical cell compare at the resting membrane potential? What are the charges like in a cell at rest?
Positive.
Positive where?
On the outer shell.
You got it. Yep. Most cells at resting membrane potential will have more positive charge outside the membrane, and just inside the membrane, they'll have kind of a layer of negative charges. So more positive outside, more negative inside. All right, it makes a gradient. Can anyone remember what the range of charges actually is if we were to measure it?
-40 to -70.
Ooh. Millivolts. So we're talking about a tiny bit of electricity here, of electrical potential there. But it's still enough to, makes enough of a difference for the cell to use it. Okay, good. And I see that happening in the chat there as well. Good. Okay, so if we could actually measure it with a probe, which has been done, tiny probe, that's the usual range for most cells. All right, the next question is, which ions are most important in establishing the resting membrane potential of cells? And so for this, I'm going to pick on Desiree's next on the list. Desiree, can you tell me at least one of the particles that's really important?
Potassium.
Potassium, yeah. Mm-hmm. because of the way all this machinery works, you tend to end up with a little potassium, a little more potassium outside the cell against that membrane. Let's see, it goes in. Right. Okay. Catherine with a C, can you tell me another important particle that helps establish this membrane?
Sodium.
Sodium is also involved, okay, to a lesser degree. Sodium sometimes will line up inside the membrane a little bit. It gets a little more complicated with sodium, but yes, good. And then there's another one, a weird one, that also is involved. Emily, can you tell me what the other weird one is?
I don't know if it's right, but an anionic protein?
Yes, yeah. An anion is anything with a negative charge, right? And so the cell will use these anionic proteins. Now, why a protein? Well, remember, proteins are big. So these proteins are going to be stuck. They are non-penetrating. They're stuck inside the cell. And since they have this little negative charge, it kind of ensures that the inside of the cell stays negative compared to the outside. Even though these other ions are moving around, these guys are not. So it helps create that gradient. Okay, good. There are a couple of other ions that are important in the process that I didn't demonstrate during the lecture and class. Can you all tell me what other ions might be involved? I already said potassium, sodium,
Calcium.
yeah, chlorine, and there was one more.
Calcium.
Calcium, 2+. Yep. So it's a balance between all of those guys. And we don't have to know exactly what each one is doing. But the membrane potential is established through a balance of all these ions. Of those, the most important ones are the potassium and the anionic proteins. All right, good. Last question here. Okay. Which proteins are most important in establishing this potential? And by this, I don't mean the anionic proteins. I mean, what machinery is used to put those ions where they need to go? And I'll just open that to the class because that's kind of confusing. So what machinery do we use to make the membrane potential?
Pumps.
Which pump?
The sodium potassium pump.
You got it. And a couple of others.
Leak channels.
You got it. Good. Y'all watch the video. I'm so happy. Yep. Sodium potassium pump is a major player. And the other two major ones are sodium and potassium leak channels that let things leak out. There are also leak channels and pumps for the other ions as well. And they all are involved, but we're not going to worry about that. Okay, sodium potassium pump and these two leak channels are the major machines that help set this up. All right, good. Excellent, y'all. Any questions about that section? All right.
There was one sentence in the notes that had two fill in the blanks. It says, in most cells, there is more blank positive outside the cell and more blank positive inside the cell. And I wasn't sure exactly what you meant because I thought it was more negative on the outside of on the inside of the cell and more positive on the outside.
There is more blank positive outside the cell and more blank positive inside the cell?
Yeah, that's what the sentence says.
Okay. Probably what I'm referring to there without looking at it. Are specific ions. So for instance, when you've got at resting membrane potential like this, like B, what we really have are two sets of, how am I saying this? What we really have are two sets of positive ions that are kind of balanced against each other. Let me see if it shows here. Okay, it shows here. So notice here that at resting membrane potential, we have a lot of potassium inside, high gradient, high concentration of potassium inside, and a high concentration of sodium outside. And they're both positively charged. So I think that's what I was looking for in that sentence.
Gotcha.
sodium outside, potassium inside because of the pump, right? But because of that and that balance of anionic proteins, the inside of the membrane actually tends to be more negatively charged than the outside.
So the sodium's on the outside, right?
Yes,
Okay, just making sure.
more sodium outside, more potassium inside. When we get into chapter 9, maybe chapter 11, trying to remember which one, we're going to talk about how cells generate electricity, like nerve cells and muscle cells. They communicate with electrical charges. And so the way that works, essentially, it's a series of steps. But basically, a channel is going to open that lets the sodium flow in, and another channel is then going to open that lets the potassium flow out. And so they're flowing in opposite directions, and that movement of charge is electricity. So this membrane potential, one of the things it allows is for cells to generate electricity. But there's other uses as well.
One thing that I was like slightly confused about, it probably doesn't matter, but like, do we create those channels and those leaks, or are they like already built in?
Yeah, good question. So let's talk about that, because that's going to kind of relate to what we're going to talk about in a minute. So those channels and those pumps are proteins. Okay. Where are the instructions for making proteins, y'all?
DNA.
DNA. Yeah, I should write it like this. So your DNA is the instructions for making proteins. Literally all DNA is a set of recipes for making certain kinds of proteins. DNA really doesn't do anything. It just holds the information. And then the rest of the DNA is switches that turn on the recipes or turn off the recipes. So absolutely, those proteins are made, these channels and pumps are made by the cell, and they'll make them as they need them. So if they make more of something, it's called upregulation. And if they stop making them or make fewer, it's called downregulation.
Is that why some like diseases are hereditary? Because it's like in our DNA?
Absolutely. Yep. If it affects any kind of a protein, which most diseases do, then it could potentially be a genetic disorder. Yeah, it could cause a disease. If it affects an enzyme that controls a chemical reaction, that's a protein as well. So that could be a genetic thing too. Yep. Excellent. Good connection. So keep that recipes for proteins thing in mind. So we're going to talk about that again briefly in a moment. All right, any other questions? Good questions today. Oh, by the way, I am still going to put up your links for your I don't know questions. It's written down on my list. Got a lot of catch up to do this week. I'll get there. Okay. Next thing I want to talk to you about is how cells use the membranes, again, membranes, to talk to one another. Communication between cells is vital. This is how your cells can act together as an organ, and how different organs can work together to accomplish something for your body. So cells have to be able to communicate, and that communication is always going to involve the membrane in some way. So first, I just want to go over some basic methods, some vocabulary of the different kinds of communication. And then we're going to go a little deeper into it and learn some of the steps and things like that. All right. So first category, direct communication. In direct communication, you have two cells actually physically touching. So a good example of this, here we have two cells that are connected by these special connectors we'll talk about called gap junctions. And this allows them to pass molecules directly from the cytoplasm of one to the cytoplasm of another. So they can share chemicals back and forth. And those chemicals can be messengers, can send messages. Another way they can do it is this way. You notice that this one cell has, I guess we'd call this a receptor. It's a protein with a special groove in it. And the other one has a molecule stuck to it that's going to act as a ligand, which is like a chemical message. And when they touch each other, when those proteins fit together, that allows communication between the two cells. Sometimes it might set off a chain reaction of things going on inside one of the cells. We'll see how that works in a little bit. But you've got two things physically touching. Great example of direct communication is with your white blood cells, your WBCs, your white blood cells. White blood cells are constantly patrolling the body looking for invaders. And the way they do that is they will check your cell's ID. So they'll come up to one of your cells and they'll say, hey, show your ID. They will bind a receptor with a molecule sticking out of your cell, the ligand. And if it fits, that tells it it's supposed to be there. And if it doesn't, that tells it it's invasive and it'll try to kill it. Or it can use these to detect certain kinds of viruses, bacteria, toxins, all kinds of things. It's always checking ID. So that's called direct communication. And they are touching.
I'm sorry.
Yeah.
Can you explain gap junctions again for me, please?
Yeah, we'll look at them in more detail in a little bit, but these are basically two little tubes that stick out of the cell. So one cell would have a tube like this, and the other cell would have another tube like this, and they connect together. And so it's like two channels that fit together, and it lets molecules go through from one side to the other.
Okay, so it's like a tunnel,
Mm-hmm.
say, instead of them actually like getting together and touching.
Yep. Yep.
Okay.
Yeah, instead of touching the membrane to membrane, they have these special tunnels that connect together. It's like two channels that connect together. Yeah. Yeah, get your badges, right? Carry your ID all the time. Body's a dangerous place. When we have white blood cells that are not reading ID properly, if they start attacking your own cells, we call that an autoimmune disease. And that's a whole big category of diseases that are caused by a miscommunication, by white blood cells thinking that one of your cells that's supposed to be there is not supposed to be there. And it causes all kinds of problems.
And isn't like too many white blood cells, doesn't that cause like cancer or something like that?
Too many white blood cells can be the result of cancer. It can cause inflammation and things like that. One of the problems with COVID, in fact, is that it tends to overactivate the immune system. And so you get this huge immune reaction, and that can actually do organ damage and things like that. Some of the worst cases are caused by too much immunity, which is a little scary. Now, when I say that, that kind of a problem is a side effect of getting the virus. That is not something that a vaccine would cause. Okay, a vaccine would actually calm down the immune system a little bit. Ooh, I feel like I just stepped off into a puddle. That's a deep one.
It's funny though, because it's like our white blood cells are as paranoid about it as we are.
Right, exactly. They're so paranoid, and when they overreact, we have problems. You get things like allergies, and you get things like diseases. If you're curious about that, look up something, and I can share a video with you guys. That particular reaction, it's called a cytokine storm. It can be a side effect of having COVID, and it can be really serious. But if you want to look up cytokine storm, and if you remind me, I can send you a video. Okay, anyway, back to what we're talking about here. Okay, so now let's talk about some of the indirect means of communication, other ways that cells can communicate. Most of the methods are going to involve spraying chemicals at one another. And so it's kind of like when I'm talking to you, sound waves are coming out of my mouth, they're going into your ears, and they're vibrating the insides of your ears. So cells do something kind of similar, but with chemicals. They actually release chemicals from one cell that can go over to another cell and combined with a receptor protein, and that'll cause some kind of response inside. So these are all variations on that, spraying chemicals at each other. When two cells are pretty close together within a tissue, we call that paracrine communication. So this guy might release this little chemical that tells its neighbor what to do. Okay, so they're not physically touching, they're communicating with chemicals. A variation on that happens with nerve cells. So this is a neuron, a nerve cell like you might find in your brain or your spinal cord. And these guys release special chemicals that are called neurotransmitters. So this is a form of paracrine communication. Sometimes it's just called neurotransmitter communication. Or another term that I really like is called synaptic. Synaptic communication, because the space between the neuron and the cell is called the synapse. So synaptic communications with neurons. And the chemical it releases is called a neurotransmitter. Okay. When these special chemicals are released into the blood, well, then they can travel a really long distance. So anytime we're dealing with the blood, we're talking about chemicals that can be sent all over the body to send messages to multiple organs at the same time. And so there's two kind of variations on that. Anytime we're dealing with blood, communication through blood, think endocrine. Endocrine. This is what your endocrine system does, is it releases chemicals into the blood that are messages. And those can travel all around the body and tell multiple organs to do things at the same time. So it's long distance communication. Okay, for instance, maybe this is a cell inside your pancreas and it releases some insulin. And the insulin goes into your blood. Maybe it travels over to the liver and it tells the liver, hey, we need some more blood sugar. Our blood sugar is getting low. Please release some sugar back into the blood. Okay, so that would be the response of that particular cell. Endocrine, blood. Oh, and when we're talking endocrine, the chemicals that are released are called hormones. Okay, hormones are released into the blood, and that's endocrine communication. Another little variation on that is called neurohormonal secretion, and that's when a neuron releases a chemical, a neurohormone, it's got its own word. When a neuron releases a chemical into the blood, we call that neurohormonal release. It's a form of endocrine communication. I think we'll just see a couple of examples of that. It's not as common. Go ahead.
I'm so sorry.
That's okay.
How do medications work? Like when they're embedded in your skin, like birth control or like the things that people put on their legs when they have diabetes?
Yeah.
Like how do those work with the cells and the secretions and all that?
Yeah. So whenever you eat a pill or something like that, it gets digested and then your intestines absorb it and puts it into the blood and it circulates around where it needs to go. Same kind of thing goes on with those patches like that. It's absorbed through the skin, it diffuses through the skin, but eventually it gets into the blood. And that's how it can travel around.
And the implants for the birth control, those are like directly into the bloodstream, right?
No.
Like it's not in my muscle, but it's like right next to my muscle. So I always wondered, like, how is that supposed to actually get out of the rod and into my body?
Yeah. It's diffusing out of that rod. It's traveling through the connective tissues until it reaches blood vessels, and then it gets into the blood.
Okay.
And travels around. Yeah, it's like reproductive organs are really far away from your arm, right? So how is it supposed to get there? Has to go through the blood.
So how does it know like how much to release? Is that just like the rate that your body absorbs it?
That's part of it. It's also just that it has a fixed dose in there. And so that chemical just kind of works its way out of that plastic at a constant rate. And it's formulated to do that. So you don't get a big burst and then none later. But yeah, it just kind of slowly diffuses at a constant rate into your tissues and into your blood.
So I think someone asked on the thing, is that neurohormone secretion?
That would be, let's see, well, it's an artificial hormone, right? But that would be just regular hormonal secretion.
Yeah, because like as you said, like it involves blood. So
Right.
right.
I'm trying to think of an example of neurohormone. The pituitary gland. There are special neurons in your pituitary gland in your brain that release things like growth hormone and there's a bunch. I'm trying to remember all of them. Prolactin, no, there's a bunch. When a neuron releases a hormone into the blood, like your pituitary gland does, that's neurohormonal. So that's different. If your brain was releasing estrogen and progesterone into the blood, we could call it neurohormonal, but it doesn't do that, right? Usually it's your ovaries and our uterus, depending on what's going on.
When women get their like periods, is that neurohormonal or is it from your like uterus and stuff?
That's ovaries and uterus. Yeah, so that would be regular hormonal. And there's organs all over the body that release hormones. So again, your pancreas, your liver, your heart even releases some hormones. So those all could be considered endocrine organs in a sense.
And chemical depression, that's whenever there's like a chemical imbalance in your brain of like certain hormones.
That could be hormonal. That could also be, a lot of that has to do with neurotransmitters.
Okay.
So brain cells talking to each other usually has been more implicated in depression, although there are some hormones that can affect that as well, and women certainly know that, right? During that monthly cycle, moods and things can definitely be affected by what the ovaries are doing, for sure. So to answer your question, that's mainly neurotransmitters. Hormones can affect that too. Yeah, that gets complicated for sure. Let's see. So that would be a hormonal secretion to transdermal. Okay. Yes. Good, y'all. Have to get it replaced. The birth control implant every 3 years. Yeah, so it sits there and releases hormones for 3 years at a time. So it's basically that particular method you're talking about is basically the birth control pill, not in a pill form. It just releases those hormones at a regular rate so that people that have them don't have to take pills every night. So it's pretty interesting, but yes, good. Not to get too off topic, but yeah, y'all, there are so many interesting methods of birth control now. There are options out there. It's amazing. Which is good. Okay, let's see. Where did I want to go with this? So yes, the next question is, if we have this little chemical that is released and it binds to a receptor, and that's outside the cell, how does that cause a response inside the cell? Well, that is conducted through a pathway that we call a signal transduction pathway. And here are the basic steps. The ligand, the signaling molecule, will bind to a receptor. That's a protein. And so it will change its conformation a little bit when that happens. And then that's going to cause a chain reaction of things to happen inside this cell. And we'll take a look at a video that shows that in a moment. But that second step is called transduction. That's where the message is getting carried into the cell. Now, eventually, those messaging molecules are going to get over to some machinery that can actually do something. And so what are we talking about when we're talking about a response? Well, it could be turning a gene on or off, gene expression. It could be activating an enzyme. It could be stimulating division or stopping division. That's an option. It could be a lot of things. It depends on what the cell is doing and what its function is and all of that. But these chain reactions, it's almost like a relay race. This guy activates another guy, that activates another guy, that activates another molecule, and eventually the machinery of the cell does something, response. Okay, that's called a signal transduction pathway. I did have a couple of videos I wanted to show you related to that, so let me see. See if I had one of them open. One moment, let me pull up the video. Ads. Yeah, okay, let me show you this little video. Boop. There we go, that's close enough. So let me know if you can't hear it. Okay, it should have sound.
Signal sent by one cell is processed and converted into a response by a series of steps known as signal transduction. Signaling molecules bind to specific receptors within the membrane. The signaling molecule bound to the receptor causes the release of a second messenger molecule into the cytoplasm. The signal is relayed through a series of molecules by chemical or conformational changes. In one type of signal transduction, the transfer of a phosphate molecule relays the signal. A cascade of phosphorylations can amplify the original signal. Eventually, the signal is converted into a response, such as activation of an enzyme within the cytoplasm or activation of a transcription factor. If a transcription factor is activated, it enters the nucleus and binds to DNA. A change in gene expression is one possible outcome of signal transduction.
Okay. So that's what signal transduction looks like. It's like a relay race. One thing leading to the next, leading to the next. Okay, let me say one more thing and then we're going to take a break because I just realized we're over break time. I'm going to have to be a lot more careful about that when we meet in person. I will be, I can do it. Okay. So some of these chemical messengers have receptors in different places. Some of the receptors will be on the membrane of the cell, and those we refer to as water-soluble chemicals, right? Now, why would the receptor be on the membrane in this case? I'll say on membrane. Well, it's because this molecule likes to mix with water. If it likes to mix with water, it is not going to like mixing with the membrane because the membrane is nonpolar, if you remember correctly. So these molecules can't cross the membrane by themselves. So the receptor for that is going to have to be outside. It's going to have to be on the membrane. We also refer to that as being extracellular. Okay. Another category of these chemical messengers are called lipid soluble. Lipid soluble molecules can go across the membrane by themselves, so they can have internal receptors. So can cross membrane. And we can call them intracellular receptors. Okay. Good examples of water-soluble guys are like insulin. Insulin's a big old protein. It can't cross the membrane by itself, so its receptors on the outside. Epinephrine's another good example. You may have heard of epinephrine. Essentially, it's a type of adrenaline. Lipid-soluble ones include any of the lipid-based things. So steroid hormones like cholesterol, cortisol, testosterone, estrogen, anything that has ster in it, is going to be a steroid hormone. Those can cross the membrane. So they have receptors inside that will kick off chain reactions. And things like thyroid hormones, T3 and T4 are thyroid hormones. You'll learn about in AMP2, but thyroxin's a version of that. Those can go across the membrane too. So the point is that these guys can go inside and these guys cannot. They're restricted to the outside. So they'll have different pathways connected to them to accomplish that. Okay, here you can see it. These lipid-soluble guys can have a receptor inside the cell. The water-soluble ones have to have receptors on the outside of the cell. Ooh, okay, I got a question there. Would a good example of signal transduction be conception? Conception itself is a little bit more complicated process because with that, you've actually got the nucleus of a sperm cell going inside the egg through the membrane. But there are signaling molecules that travel with that little nucleus from the sperm that set off a whole huge cascade of signal transduction pathways. So the sperm fusing with an egg itself is not technically signal transduction, but the molecules traveling with it set off a whole bunch of those pathways. So yes and no, yes and no. Okay. All right, now it's time for a break. I was having a good time talking, got all excited. Let's take a break for about 10 minutes. We'll come back and work on this a little bit more. And then we'll jump over to chapter 1 and see if we can get a chunk of that done as well. So it's 1:31. Let's meet back here at 1:41. Good work today, guys. That was quick. Oh, let someone in. All right, y'all, welcome back. Okay, so hope you had a good break. Got up, did something else for a few minutes. Let's continue with chapter 3. And I need to speed up a little bit, but I have an activity for you guys to think about with this as well. Okay, so we left off by talking about receptors that are lipid soluble that have intracellular receptors. And we were talking about water soluble molecules that cannot cross the membrane by themselves. So they have extracellular receptors and they will connect to these long, complicated signal transduction pathways. Next, let's talk about the different effects these guys can have, the different things you can accomplish with these different kinds of receptors. Okay, so first we'll talk about the water soluble messengers. Again, they're going to have receptors on the outside of cells, extracellular. One form of this is with chemically gated ion channels or chemically gated channels that we talked about a little bit earlier in the chapter. So again, the way that this works is that this channel, this tube through the membrane has a little gate, and when the signaling molecule binds to it, which we call a ligand, it causes it to open, and that's going to allow something to flow through. Now notice that the chemical that flows through it is going to be different than the chemical that binds to it. So the messenger is typically different than the chemical that flows through, although it depends. Okay, so one thing that these messengers can do is open a channel, cause things to flow. Another thing that they can do is to activate enzymes. So in this case, we have a ligand binding to a receptor. That's going to cause that protein to change its shape, which is then going to cause another protein to change its shape and activate. And then that enzyme can go out and control a chemical reaction. If you remember, enzymes catalyze chem reactions. That's what they do. They catalyze chemical reactions. They speed up chemical reactions. So you can have this ligand signaling an enzyme to do a job. That's another option. Another thing that these water-soluble messages can do is kick off what's known as a G protein pathway. And there are a bunch of steps to this, which we're not really going to go through. But what I want you to know is that G protein pathways are extremely common in cells. I think they found that something like 90% of medications affect these G protein pathways in some way or another, affect some component of a G protein pathway. So all cells are going to have G protein pathways, and they can be connected to lots of different machinery to cause lots of different responses. But the way they kind of work is that the ligand is going to come down and bind to a receptor. That is then going to cause that receptor to change its conformation, its shape. And then it's going to activate this guy, which is called a G protein. The G protein will travel inside the cell until it hooks up with sometimes something called a secondary messenger. And there's a whole list of secondary messengers, but the one that I really want you to be familiar with are this guy, cyclic AMP, CAMP. That molecule serves as a secondary messenger in lots of these pathways. And there's another category called kinases, protein kinases, and they are often involved in these pathways as well. Okay, so they would be kind of over here. And then those secondary messengers are eventually going to result in some kind of response. Okay, so again, it's kind of a relay race, kind of an assembly line. And these water-soluble messengers can kick those off. There's more information on it if you want, but that's as far as we're really going to go with it. As far as G proteins, we'll see examples later. All right, now the lipid-soluble chemicals, they behave a little differently than the water-soluble ones. So let's take a look at that. Here, for instance, we have a chemical traveling through the blood like estrogen or testosterone. Here it's labeled a hormone. And so because it is lipid soluble, it's going to be able to come into the cell through the membrane. And eventually, once it's inside the cell, it's going to find its receptor. So it's an intracellular receptor. Now, these intracellular receptors can be connected to lots of different kinds of machines in the cell, and it just depends on what receptor and what hormone or what chemical you're talking about. But one thing that it can do that's kind of unique is that since these receptors are intracellular, oftentimes these things will travel into the nucleus and actually interact with the DNA. So these guys can sometimes act as switches to turn genes on or off. And so we say that they affect gene expression. Now, I'm saying that they affect gene expression. Oop. If they affect gene expression, turning a piece of DNA on or off, what are they ultimately going to affect? What are genes recipes for?
DNA.
They're part of DNA.
Proteins.
Proteins, yeah. They affect which proteins are made at which times. So by affecting gene expression, the DNA, it's going to determine through a series of steps which proteins, here it is over here, which proteins get made at which times. And different proteins have different jobs. So it affects the workforce that's present in the cell, so it can affect how the cell behaves. Okay. And usually when we talk about molecules, signaling molecules that affect gene expression, we're talking about something that's lipid soluble. Okay. And there's some other things. Since I'm talking, let me talk through this last little section here. This is another topic, but let me talk for just a couple of minutes about this, and then we're going to do an activity. okay, to kind of reinforce all of this stuff. So in this last section, we're going to talk about some special connectors that connect cells together. There's some special structures that you might hear about. And there's 3 kinds of connectors that I want you to be familiar with. Okay, in this diagram, this is kind of cool. This cell has been hollowed out. So the perspective is that if you were here standing inside this cell, looking at the membrane from the inside, and you can see how it's connected to the next cell over. Okay, so the first type of connector I want to talk about are these guys. These are called tight junctions. Tight junctions. Tight junctions always kind of remind me of the hem on a shirt. You know, you have that little line of stitching along the bottom of your shirt or along the sleeves on a t-shirt. That's kind of what this is like. These proteins weave back and forth through the membranes of 2 cells that are next to each other, and it stitches them together. This provides a really good seal between the 2 membranes. And so we see these tight junctions a lot in cells that make layers in the body. So like epithelial tissues that make barriers, or the kind of skins on the outside of organs, we see a lot of these tight junctions there. And it gives the cell a lot of control over what passes through the cells and what can go between them. Okay, so those are tight junctions, stitching them together.
So then are those able to be unraveled by like just the usual protein denaturing things like heat and all, or is there something like special about them other than that?
Yeah, they can be affected by heat and by different denaturing chemicals like that, like acids and bases, along with all the other proteins. Yeah, they could be pulled apart that way, or just by physical force, they could be pulled apart as well. Yeah, good. Okay. The second type of connectors that I want you to know about are called desmosomes. Desmosomes. And for desmosomes, I want you to think about it being like a bolt. So like here we have 2 cells and we're using a desmosome to bolt them together. These junctions are really, really strong. And they're made out of proteins called cytoskeletal proteins. So these are parts of the cell skeleton that extend from one cell to the next and stitch them together, bolt them together. And we see these kinds of connectors in cells that have to take a lot of force. So we see them connecting heart cells together, for instance, and cells within your muscles have lots of desmosomes. And we also see them in some of the upper layers of your skin. Your skin needs to be really tough, right? And glued together to make a nice barrier. So we see lots of desmosomes there too. They're bolted together.
So another question.
Sure.
Do you know if there are any diseases that specifically affect these connective proteins that make cells not sealed together as well or anything like that?
I don't know of any diseases that specifically affect the desmosomes or the tight junctions. There are diseases that can affect like the connective tissues and things that connect things together on kind of a larger level. That's a good question.
I'm kind of curious about the cellular level because it seems like if it happens on a bigger level, then maybe it could happen on a small level. I don't know.
Both could cause problems. Yeah. If you have a tissue in your body that's supposed to separate 2 things, 2 areas, and the cells are not sealed together properly, that could cause big problems. But let's see. Let's make that one more. You guys now have 3 I don't know questions, so you have a choice. Which ones you want to do? Let's see.
I have one quick question. I just think about when he's talking about denaturing proteins. So if you were to physically like poke a cell, would the intracellular fluid like spill out, or would it be more like a gel matrix?
It's enough of a fluid to spill, so it can come out. It's a little bit more of a jelly than just like water.
Okay, I was just always kind of curious about that.
Yeah. Let's Just trying to think of an example of doing that. Because when you cut something apart, like you slice a tomato and all the liquid comes out, that would be like interstitial liquid between the cells that you were mostly seeing. But some of that would be intracellular fluid as well. So it's fluidy enough to flow, but it is a little bit more jelly-like than just pure water or something. Yeah, let's see. Are there any diseases that?
So like cold syrup.
Uh-huh. Go ahead. By problems, malformed, denatured. There we go. You can hear all my typing noises there. Are there any diseases that are caused by malformed or denatured desmosomes or tight junctions? Let's make that I don't know question. And I really will put up the links for these so you guys can answer them. I'm gonna take a picture so I remember to do it.
And more than one person can answer them, right?
Oh yeah, yeah, this is open to anybody who wants to research it. Just make sure you provide a source with it. And you can answer 2 for credit.
And I would suggest if you're looking for a source to check the ACC library website, because they usually have really good sources that are applicable.
That are legit, yeah, as opposed to just Google.
Exactly.
Yep, good advice. Yeah, we have an awesome library.
Having that journal access really does help out with research.
Mm-hmm. Yeah, it's really good, and our librarians are so knowledgeable. They're really amazing. Was there another question about these? Okay, so let's find out. That would be really interesting to know. All right, the last type of connector that I want you to be aware of, I kind of already talked about, so I'll be really brief here, but these are gap junctions. And gap junctions are like having 2 transport proteins stuck together. So you can see here that the blue, this is one cell membrane, this is one cell, and over here is another cell. And you've got these tubes that stick out that connect them. So it would be kind of like this. Here we have one membrane, here we have another membrane, and this one has a gap junction that sticks out, and this one has a second one that sticks out, and they actually connect together. These are a little bit more complex than gated transport proteins or gated channels because they're actually made out of a bunch of subunits, and they can twist to open and close. So this gives the cell a lot of control over what it shares with its neighbor. And down here you can see the little gap junctions. These tubes allow cells to share chemicals directly back and forth. We see these a lot they're all over the body, but we see these a lot in cells that have to work together as a team really, really precisely. So a great example is heart cells. This is part of what helps heart cells contract as a unit. Part of that. Okay. So gap junctions are really, really cool. But if you hear about them, just think about they're just connections. Junctions across a gap. That's what they are. Okay. Oh, we made it, guys. We made it to the end of chapter 3. I never thought it would happen. No, it's a long chapter. Questions about any of that?
This might be a little off topic, but I was just curious, what are we going to be dissecting when we do the dissections?
When we get to that, we're going to do a brain, a sheep brain, and a cow eye.
Oh.
Yeah. Brains and eyes. Let's see, where are we at here? Here.
Is that going to be around Halloween?
That would be awesome. I will say that we're going to get into the skeleton not too long from now. And this is a perfect time of year for skeletons because there's skeletons in the stores and there's also brains in the stores. Now, they're not really great models, but they are something that you could work with, right? So I'll remind you of that when we get closer to time. Okay, so what we're going to do is go back to this worksheet And I've got activity 3 here. And so activity 3 is going to give you a couple of scenarios asking you what kind of receptors or what kind of chemical signaling molecules you think are involved here. So I want you to work together to see what you can answer there. And then if you scroll down below to the next section, you're going to see that it gives you 3 scenarios, and it's going to ask you what kind of signaling you think is going on there. Okay, so I would like you to do activity 3 on the worksheet next. Okay, so we have this one about nerve growth factor. That's a chemical. And it says it's water soluble signaling molecule. So based on that, would y'all say that the receptor is extracellular or intracellular?
We should
Yeah, go ahead.
Take a stab at it. I'm thinking it's extracellular.
Mm-hmm. Why?
Kind of what I was thinking. Because of the membrane being what's the word I'm looking for?
non-polar, hydrophobic.
Hydrophobic.
Mm-hmm. You're correct. So if a molecule is water soluble, is it going to be, that means it mixes with water, right? So what does that mean? It's hydrophilic or hydrophobic?
Hydrophilic.
Hydrophilic. If it's hydrophilic, will it be lipophilic or lipophobic? Lipophobic, right. And so given that, since it's lipophobic, it's not going to want to cross the membrane by itself. So that means that the receptor for it would have to be on the outside. You got it. So that's a extracellular receptor. This one. Okay, so you're told that these chemicals, you're given more information than you need, but you're told that the chemicals are lipophilic. So group 2, do you all agree with this, that the receptor would be intracellular? Someone from group 2. Yes?
Yeah.
Since it's lipophilic, it will also be hydro what? Phobic. So that means that it can cross the membrane. So it can have an internal intracellular receptor. Good. Okay, let's talk about these. So, and let me see what we're looking at here. Let's jump over here to group 3. Okay, so this first one talks about heart cells releasing a chemical into the plasma when they are overstretched. Group 3, you said that's an endocrine communication. Why did you say endocrine? What keyed you into that?
That plasma has to do with blood, and endocrine releases into the bloodstream.
Yep. If it goes into the blood, that's going to be an endocrine signaling mechanism. Good. All right. Number 2 was talking about beta endorphins are released in between neurons in the brain. These chemicals, blah, blah, blah. Okay. Endorphins help numb pain in the body, which is kind of cool. But group 3 here, you're saying that that was synaptic signaling. What tells you that it's synaptic? What clue was there?
Came from the brain.
Brain. And it also says.
And the fact that it like jumped is what clued me in.
Jumped where?
Let me find it again.
Across the space between neurons.
Yeah.
Yeah, you got it. Between neurons is the clue there. Synaptic signaling happens between neurons. You got it. All right, group 4, I'm going to pick on y'all a little bit here. Let's see what y'all put for this. Okay, so for this third one, I said, okay, cells infected by viral particles release something called interferon into the extracellular environment, and it travels between the cells, neighboring cells, and binds to receptors, and it helps protect them against infection. So y'all said paracrine. Why did you say paracrine? Group 4.
Oh, to be honest, we didn't get to this part.
Oh, okay.
I didn't really know, but it says that it's talking about neighboring cells, and since it's local.
You got it.
Because it released into the extracellular fluid.
Yes, you got it. You got it. So between neighboring cells, that's going to be called paracrine, or local is another term for that you see sometimes, but we're going to use the term paracrine. All right. And then the last one here, I'll just open up to everybody here, but let's see, we've got number 4, immune cells, such as macrophages, antigen presenting cells, APCs, and helper T cells. Pull antigen molecules off of membranes of invading particles and present them to one another, add them to their own. What do y'all think for that one? What kind of signaling would be involved in that whole process?
Direct, because it involves physically touching.
You got it. Excellent. That's, your immune system depends a lot on direct signaling. So for instance, when you get a vaccine, there's different vaccines that work different ways, but part of what happens is that those molecules that are injected into you act as antigens. They act as little signals, and your immune system will pick those up. And there are cells in your immune system that will stick them on themselves, and it helps them identify those viruses when they do get into your body. So the vaccine makes your immune system react faster to that particular virus. That's a very simplified explanation, but direct signaling is involved there.
You did say that these will be on Blackboard.
Yeah, absolutely. Mm-hmm. Okay. Any questions about chapter 3? Goodness gracious. It's such an important chapter for your understanding later on. Again, especially A and P2. Okay. What I'd like to do next is switch topics and jump over to chapter 1. And let's talk about some chapter 1 stuff. Now, some of the stuff in chapter 1 we've already talked about. So I'm not going to go over everything that's in here. But there are a couple of topics that can be confusing, so I want to make sure we get to that. And then eventually I want to be able to get to the lab stuff. We may end up doing it next time, which is fine. What am I looking for? My chat. Where did it go? Here we go. All right, I got my chat pulled up in case you guys have questions. Okay. So this chapter, I kind of move it around different semesters, just depending on, just kind of how I'm feeling as far as where it fits in. But technically, it would probably be better to do this chapter after we talk about chapter 4. Because in chapter 4, we're going to talk about how cells work together in groups that we call tissues. But there's some good introductory stuff here that fits in with what we've been talking about. So we'll tackle those topics. All right. Here we go. The major chapter 1 topic I want to talk to you about today is something called homeostasis. Let's see if I can get that out of the way. There we go. Okay. And so this is an important introductory topic because it talks about how the body is able to achieve equilibrium or to achieve a certain amount of balance so that you stay alive from one moment to the next. Maintaining homeostasis is about maintaining a stable internal environment, despite the fact that the external environment is changing constantly. Okay. Body temperature is a great example of that. It's our classic example. It's pretty nice today, but often in the summertime in Texas, if you go outside, it's going to be really hot out there. And so the challenge for your body is to maintain a stable body temperature, right? 98.6 Fahrenheit, even though it's 106 or 115 outside. Or if you go out on a really cold day, keeping your body at that same temperature or close to it, even though it's 40 or 50 degrees outside, or even colder, if you think about last February. So homeostasis is the art of doing that, of keeping an internal environment that's pretty stable, despite the fact that the outside environment changes all the time. Okay.
So like shivering to warm up would be an example of like us trying to maintain homeostasis.
Yes. Yep, shivering or sweating are both your body trying to maintain homeostasis. Now, things are constantly changing within your body within narrow limits, right? Think about all the factors that can change in your body. You've got different chemical concentrations, you've got fluids moving around, you've got electrical events, you've got heat moving around, chemical reactions happening, all of that stuff all at the same time. And so the challenge is for your body to keep things fairly balanced. So I think of maintaining homeostasis kind of like this waiter. If you've ever tried to carry a big tray with a bunch of stuff on it, you know that you have to constantly adjust to keep that tray balanced, right? It doesn't just stay still. And if things get a little too out of balance, things can start spilling and the tray will rock back and forth wildly and you drop stuff. That's not a good situation in the body. But on a normal day, from moment to moment, you have to rock the tray to keep it balanced. That's homeostasis. So now what are we talking about when we talk about maintaining this internal environment? Well, again, I would encourage you guys to think of the internal environment not as a bunch of organs put together, but rather as these reservoirs of fluids that interact with each other. Because our bodies essentially are made of 3 different reservoirs of fluid. Or 2 different really, with a subdivision. One of them is the ICF, the intracellular fluid. And so that's just that liquidy stuff inside cells. And so most of your body's water is stored there inside the cells. And then around the outside of the cells, of course, we have the ECF. And that's another pocket of fluid. The ECF, again, can be broken down into 2 different types of fluid. The liquid that's between the cells within a tissue, we call interstitial, also abbreviated IF, or I've seen ISF as well. And then the fluid that's inside the blood, like that particular fluid I showed you on the last slide. This is blood right here. There's a white blood cell and a red blood cell. That particular kind of ECF would be called plasma. So it's kind of a weird way to think about the body as being these kind of bags of fluid that interact with each other, but that's true. It's true. It's a little bit more complex way of thinking about the body. But basically, in homeostasis, we have the interaction of these 3 reservoirs of fluid, and we constantly have chemicals moving back and forth between the plasma and the interstitial fluid around the cells, and between the interstitial fluid and the intracellular fluid back and forth. It's the interaction of those fluids with each other that keeps us stable and keeps us balanced. So those are the internal environments. Okay. Let's think just for a moment about the body as a whole here. Let's zoom out a little bit and think about that. How do I want to draw this? So let's say that we have these cells. These are some cells within our tissues of our body. These cells contain the intracellular fluid. And then around the outside of them, we have the interstitial fluid, right? We are going to need to get to these cells nutrients and oxygen. And we're going to need to get out of these cells things like waste. So how does the body accomplish this? How does the body maintain these 3 major reservoirs on a larger level? Well, let's start by looking at how it gets nutrients in. Y'all know this. Where do you get nutrients from?
ATP.
That's where you get energy from.
Food.
Food, right? Okay, this is your digestive system. Dig, sis. Mouth. Anus. Mouth at one end, anus at the other end. All right. So there is going to be a transfer between the digestive system, the cells of the digestive system, and the interstitial fluid of the tissues. Okay, we have nutrients that are coming in, we have waste products that are going out ultimately of the body. So we need to be able to get those nutrients into that intracellular fluid and into the interstitial fluid. Another place we're going to have an entry and an exit is the lungs. These are lungs. Okay, what are we going to take in through the lungs, y'all? That's kind of important.
Oxygen.
Oxygen. And what are we going to get rid of through the lungs?
CO2.
CO2, yeah. CO2 is a waste product, so we get rid of waste through our lungs as well. So we need to be able to get this O2 and CO2 to those cells as well. So the big question is, how the heck do we do that? Well, it's that third reservoir, the plasma, that connects this all together. Okay, here's a heart, here's blood vessels. Okay, what will happen is that food and nutrients are going to be collected by the intestines, and they're going to be placed into the plasma by some of the mechanisms we've already talked about, by moving things across cell membranes. All right, and then from the plasma, it can travel into the interstitial fluid and into the intracellular fluid. You're going to have waste products going the other way as well. Okay, some of that waste actually will get placed back in the intestine and can be pooped out. But other waste will travel from the blood to the lungs, like CO2, to get breathed out. And oxygen is going to be moving the opposite way. Okay, so the circulatory system is really the system that connects all three of these reservoirs together. So the plasma is a really important fluid reservoir. And when we talk about maintaining homeostasis, we're really talking about the balance between all three of these fluids. The lymphatic system plays a part too. We can talk about that later. Okay, now, homeostasis is all about maintaining what we call variables. And a variable is anything that can change in the body. So temperature, chemical concentrations, body position, all of those things can be variables. For each variable, we have kind of a goal that the body tries to set, or a level at which it tries to maintain a certain variable, and that's called a set point. So for instance, y'all, what would the set point be for body temperature?
98.6.
Anyone know what it is in Celsius?
.36 Sorry, 32.
Oh, 37.
Sorry.
37, you were close to the percent, yeah. 37 degrees Celsius or 98.6 Fahrenheit, right? So that would be our goal here, the ideal, let's say 37 degrees Celsius. But for each of these variables, we are going to have a maximum that we can tolerate and a minimum that we can tolerate. I think the maximum, if I remember correct, oh wait, let me say 98.6 degrees Fahrenheit, since these are in Fahrenheit. I think the maximum that we can tolerate is somewhere around 104, 105. And the minimum, I believe, before you become hypothermic is somewhere around 95. So we really have a pretty narrow range of degrees that we can tolerate. And the challenge for the body is to keep things at around 98.6.
Why is it that children and like babies can run a higher temperature than adults? Do you know?
Let me think about that. It has a lot to do with their metabolism. Their metabolism is really high, so they tend to release more body heat.
Is that because they're smaller in size? Like, you know how bigger animals, we need more food to eat, therefore we like give off less heat, and then with tiny animals, they need more energy?
That's that might be part of it. It's mostly that they're growing. Right? Their bodies are growing constantly. So there's tons of things like cell division going on that we don't have as much of. So they burn hot. They take in a lot of food, burn a lot of sugars, make a lot of heat. Okay. Here we have an example of blood calcium levels. Yes, blood calcium levels that are, we have a range that we can tolerate. And you can see that it's actually a fairly narrow range. And then within that, your body's going to have an ideal set point for the amount of calcium it wants to have in the blood at any given time. Okay, and that's going to be true for any chemical, any ion that you think about. There's a set point and then a range we can tolerate if it starts to get a little high or a little low. Okay, this graph just illustrates that these factors, these variables, do not stay constant within a day. The goal, again, is just to kind of be near that set point and not so high or so low as to kill us. So this graph is kind of cool. It shows how your body temperature tends to fluctuate during an average day. Now, if you work overnights or you have a different schedule than this, your chart might look a little different. But in general, body temperature tends to be lowest when you first wake up. That's because you've been laying still. Your metabolism's really low. Tends to rev up through the day, peak out around 4 p.m.-ish, and then going into the evening, it tends to lower. This can be because of activity level. Of course, you might have a spike. If you're exercising, your temperature might go up, or if you go outside and it's really hot, that might spike and change a little bit. But the point is that it can change through the day. As long as you don't get too hot or too cold, you're going to be fine. You have maintained homeostasis. Now, when we get a factor that is out of whack, outside the range, or is fluctuating a lot within the range, we refer to that as a homeostatic imbalance. So technically, all diseases and disorders can be referred to as a homeostatic imbalance. And I think that's how your textbook refers to them. It'll say homeostatic imbalance, and then it'll tell you about a bunch of diseases. So those sections are kind of interesting. But it's some imbalance with a variable in the body. Here you can see blood glucose levels of someone with normal blood glucose regulation. You can see it does go up and down through the day, but within kind of a normal range. And you can compare that to someone with diabetes. Diabetes is the inability to control blood sugar, the amount of sugar in your blood, amount of glucose in your blood. So you can see that every time this person eats, they get a big spike, and then they get a huge drop in blood sugar. It's not being regulated well within the normal range at all. So it goes up and down. That's really tough on the body. That can be really hard on the organs in a number of ways. It does affect the blood's ability to keep the right levels of water and other ions and all kinds of things. So it's not a good thing, not a good thing. That's a good illustration of a disorder that takes something outside of its normal range.
And that's
imbalance.
Of like the inherited.
It can be.
What's the difference between the one that is like inherited and the one that is based on like situations?
Yeah, it's, let's see, type 1 and type 2, and I tend to get these backwards. Okay, some of y'all will know about this.
I have type 2 diabetes, and it's well, as I have genetics too. My mom's on my mom's side and my dad, so I just got it from both of them.
Oh man.
Yeah, but then I think type 1 is where you're born with it, where you don't have the pancreas is not giving up the insulin or the bringing down your sugar. But yeah, I just found out I was diabetic like in May.
Oh, I'm sorry to hear that. I'm sorry to hear that.
I was almost in a coma and my sugar was like 2,400.
Ooh, mercy. Well, I'm glad they caught it, and I'm glad you weren't in a coma. Goodness gracious.
Yeah, so I've definitely spiked just like that. It's crazy.
So okay, thank you for clarifying that. I always get those mixed up, but type 2 is the one that's more affected by diet, and type 1 is a little bit more of a specialized thing. But yeah, it's going to have a big effect on the body for sure. Okay, So the ability to maintain homeostasis is going to be controlled by two systems. One, this word should look familiar, endocrine. This is the endocrine system. It's a series of glands in the body that release hormones, and the hormones help the whole body communicate with one another. All the organs communicate. So it's about communication. And the other major one is the nervous system. The nervous system connects the organs together with nerves, and it uses electrical impulses to allow the organs to communicate. So these systems are all about communication. Letting the organs talk to one another. These are the two major systems that help oversee this whole process. Okay. Okay, good. Couple more things to know about homeostasis. Yeah. One is that homeostasis is maintained through a series of what we call pathways. Pathways involve the taking in of sensory information, thinking about it, and then deciding what to do about it. So it's kind of a decision tree, if you want to think of it that way. So let's use this as an example. Let's say that you are thirsty. Okay, this guy is looking at this water. He takes in that sensory information. There's water there. I'm thinking about my body and my the amount of water in my blood, all of that stuff. That information is going to travel into the body along what we call an afferent pathway. Af means in. Okay, it's taking information in. It's going to travel somewhere in the body known as a control center. And usually we think of the brain or the spinal cord as being the control center, but it can be other parts of the body too, depending on the pathway we're talking about. But the control center is going to think about it. It's going to say, hey, there's some water there, and we could use some more water in our blood or in our tissues. We should probably get that water. The control center is then going to send out another impulse out along what we call the efferent, think E for exit, efferent pathway. And it's going to go to another part of the body that can do something about it. The part of the body that can do something about it is called an effector. There's the word effector. It will result in the effect, right? In this case, it's the arm muscles, and the arm muscles are going to grab that water and put it to the face and allow the person to drink. Okay, so we thought about the environment. The information went in along an afferent pathway to a control center. Control center thought about it and decided, hey, we need that water. So it sent a second impulse along an efferent pathway to the arm, and the arm is going to do something about it. It's going to respond. It's called an effector. Let's get that water. Now these pathways occur, you have thousands and thousands of these pathways occurring for each of your variables all the time, and they double check one another, which is kind of nice. So once the person drinks that water, then the pathway is going to happen again. He's going to think, okay, was that enough water? Make a decision. Too much water, not enough, whatever. It'll think about it and do it again, over and over and over again. Okay. That should also be underlined. I think my picture's covering it. Now, homeostatic pathways can respond to a stimulus in a couple of different ways. In other words, there's a couple of different kinds of homeostatic pathways. And there's several different ways to think about these, but I'll tell you how I think about them. Okay, the thing I like to do to identify which kind of feedback I've got going on, which kind of pathway I've got, is to look at the stimulus. So what's stimulating all of this? If the reaction to the stimulus is trying to increase it, make it greater, we call that positive feedback. If, on the other hand, I am trying to decrease the stimulus, make it less, then we call that negative feedback. Okay, these terms have nothing to do with something being a good reaction or good for the body or bad for the body. So don't think of positive and negative as good and bad. Positive increases the stimulus, negative decreases the stimulus. Okay, my silly example here is with this mouse. And so if we think the mouse eats some cheese and the signals that it has eaten cheese goes to its brain and it goes, oh, that's really good, I'm going to keep eating cheese. That would be a positive feedback loop. Keep eating more. On the other hand, if it encounters something horrible like a cat, it's going to want to decrease that stimulus. It wants to get away from that thing. So that would be a negative feedback. The actions it is taking now represent a negative feedback system.
So, quick question, what's it called when like something interrupts the pathway of a signal? Like, for example, like there's this one kid at my school who he like is hungry and he wants to eat. He just doesn't like take that step and initiative to like eat the food.
Okay, so the signal itself has been interrupted. Trying to think if there's a specific term for that. That would definitely be a signaling problem, but I think that's different than feedback. So if you're talking about eating, right, the hunger impulse, that mouse being hungry, would actually be negative feedback, because the hunger is something it wants to do away with. So it would eat the cheese in an attempt to get rid of that hunger. So the hunger impulse would actually be a negative feedback system. So there's just something interrupting that feedback system for that person. And hormones can serve a lot of those signaling molecules too. So there's like hormones that make you feel satisfied with a meal, that kind of thing. But I don't know the specific term that you're referring to with that. Let's look at this pathway quickly here, though. What kind of feedback would this pathway represent, y'all? Think about what the stimulus is,
Positive.
what the reaction is.
Positive, because
Positive?
it's, and it's I need more of that.
So what would the stimulus be if you're looking at some water and thinking about drinking it?
Oh, it would be that you were
Mouse
thirsty.
dry.
Thirst would be the stimulus, right? So the action of drinking reduced the thirst. So that would be a negative feedback.
So aren't most things both though? Because like it's the I see it and we need more of that, but at the same time it's like I don't want to be thirsty.
Well, if in that case, if this was a positive feedback loop, then drinking the water would actually increase the thirst. And so they would want to drink more water and more water and more water.
Okay, so it's like you already have to like get it in the first place to want more of it, sort of.
Right, right.
Well, if this is an example of negative feedback, I don't understand how the mouse and the cheese, how that is what that was.
Okay, maybe that was a bad example.
Well, the mouse is already eating the cheese.
Right. The idea was that the mouse tastes the cheese, and the taste is the stimulus. So not hunger necessarily, but the taste in that case would be the stimulus. And then that would tell the mouse, that's really good. I need to eat more and more and more of it. Let's look at a couple of different examples. That might just be not a great analogy. Okay, let's think about, does it say it on here? I don't know. Let's think about regulating body temperature. If your body temperature starts to go up, start to get hot, your body will respond by sweating, right? You're sweating. And that helps cool your body down a little bit. So we had something going up, and now it's coming back down. On the other hand, if your body temperature starts going down, you start getting cold, you might shiver, and the shivering helps warm you up. So your body temperature starts to come back up. So it's kind of a seesaw going up and down. Would this represent positive or negative feedback?
Negative or positive?
One of them. So the stimulus, for instance, here was the temperature's going up and an action was taken that brings it back down.
That would be negative.
That's negative, yeah. And then if you start to get cold, that means your temperature is going down and the body took an action to bring it back up. That's negative. Negative feedback system. If we were to graph a variable and the changes in a variable, this is what it would look like if we had a negative feedback system. So negative feedback systems will allow the variable to go up and down, but it's always going to rocket back and forth around a set point. So it never gets too high or too low. So we say that negative feedback is auto-regulating. It kind of regulates itself.
So could you say that, like, if you have a negative feedback, would that be like your body feels negatively about something, like the cold or like being hungry? And then the positive, is it like responding positively, like tasting the cheese and liking it?
I wouldn't try to remember it that way, just because it's not about whether it's good or bad. It's about whether you're going towards a stimulus or whether you're trying to stop the stimulus. Because sometimes positive feedback can be a lot harder to control than negative feedback. It doesn't control itself. It has to be manually shut down. So in that way, positive feedback often gets out of control and can be quote unquote bad, right? So I don't think I'd think of it in terms of good or bad. It's think about what the stimulus is and whether we're trying to reverse it or not. Okay, here's an example of positive feedback. Maybe this will help. So whenever you get an injury, there are going to be chemicals that are released that will bring white blood cells to the area. Okay. Now, when the white blood cell arrives, it is going to release chemicals into the blood that attract more white blood cells. So this white blood cell releases a chemical. That's the stimulus. Okay. When it arrives, it's going to release more chemicals that recruit more white blood cells. So some others are going to show up. And then they are going to release chemicals that bring even more. So pretty quickly, you can get this army of white blood cells that arrive at an area of injury in response to that first white blood cell releasing its attractive chemicals. So the stimulus really in this case is the white blood cell releasing the chemicals. Okay. And it's trying to attract more and more and more white blood cells to that area. So that's an example of positive feedback because the stimulus keeps getting bigger, bigger, bigger. And the variable is changing in one direction. Okay. For instance, if you have one white blood cell down here that starts releasing chemicals, as time goes by, you're going to get more and more doing the same thing until at a certain point, you're going to reach what's called a climax event. Okay, and at that point, there's going to be some other factor that comes along and turns off this reaction. It's going to say, hey, calm down. We got it. You cleared out all the bacteria. You're doing a good job. And then the reaction will start to calm down again. So the difference is that the variable doesn't rock up and down. The variable in question increases rapidly up to a certain point and then has to get shut off by a second mechanism. So that's positive feedback. Another good example of positive feedback is childbirth. Contractions during childbirth. Oh, I just realized I'm over time. Okay, contractions start out small. The pressure on the uterus encourages contractions to happen, and they get bigger and bigger and bigger until the baby is released. It just flew off into space there. And then the removal of the baby from the uterus is what will, that second event is what will allow the muscles of the uterus to calm down. Okay, that's positive feedback. Okay, most of the time your body uses negative rather than positive because negative is auto-regulating, whereas positive has to be shut off manually. Okay, we'll play with that some more next time because I know that those feedback mechanisms can be kind of confusing. I may have explained it in a confusing way. I hope not. Okay, sorry about that. I just got to talking. All right, y'all. So we'll continue with chapter 1 next time. I've got another activity for you to practice with, and I can give you some more examples. We can suss this out a little bit more. I will send out an email with the instructions on how to get ready for next time. Next time we're going to work on chapter 1 and hopefully start a little, we'll do some in-lab activities. Very exciting. We're going to meet in person. Anyone have any questions at the moment?
Which lecture do we need to watch before next time?
I'm going to take a look at that in a moment and I'll send it out in an email. Let me look.
I had a question about the exam. Is it gonna, it's just chapter 1, 2, and 3, right?
Yeah.
That's the material on there?
Yep. Chapters 1 through 3.
And then whenever you, is it like 1.6 or whatever the numbers were for the sections in reading, is it just gonna be what's over that part in the chapter or all of it? Like
yes. Yes. If there's extra stuff in the textbook that we didn't go over, then we're not gonna, it won't be on the test.
Okay.
When's the 1 through 3 test?
I'm sorry?
When's the chapters 1 through 3 test?
It's not next week, but the following week.
I think it's the 21st of September.
I think so. I don't have it in my head, but check the course schedule.
It's the 21st of September, a Tuesday.
Um,
All right. Okay. Well, thank you for sticking with me. I had a class let me just keep talking for a good 15-20 minutes one time before someone said class is over. So at least I noticed this time, y'all. I'll see you guys next Tuesday in person and at Cypress, and look for my emails because I'm going to give you lots of instructions about what to expect, etc. Okay, questions for me?
Thanks so much.
Yeah, y'all have a good weekend.
You too.
Bye.
see you for real next week.
I know, it's exciting.